I'll make a short introduction of our speaker of today. Is Eleni Psarou. Elena studied psychology in Padua University, graduated on 2014. Then she pursued her master in brain and mind sciences and graduated in 2017. She holds a PhD from Donders Institute for Brain, Cognition and Behavior in the Netherlands, and currently she's a postdoctoral fellow at Zero Noise Lab, Ernst Strüngmann Institute of the Max Planck Society. Her research focuses on the role of neuronal oscillations in primate cognition, and this is particularly interested in the new opportunities that the virtual reality offers for cognitive neuroscience. Elena, thank you for accepting our invitation. It's a great pleasure to have you here. The floor is yours.
Hello, thanks for the invitation and for the introduction. Yeah, I'm very happy to be here again, back to the graduate program. So I will present today a small part of my work, of the work I have done since I graduated from the Brain Mind program. Yes, okay. So I will start with a short introduction to my background, even though already Vasilis told most of the things. And then I will talk a little bit about my experience in the Brain Mind Masters, and then I'll talk about my research on neuronal oscillations. And finally, I will end up with the slides on what I would tell myself if I could go back in time before starting a PhD. Feel free to interrupt me and ask questions during the talk. And yeah, so I have a bachelor in psychology, and during my bachelor, I actually completed an internship on neuropsychological rehabilitations for patients with traumatic brain injuries. And during this internship, I realized that we actually, if we want to treat the brain disease, we actually need to advance our knowledge about brain physiology, which then motivated me to turn into basic neuroscience, and that's how I ended up applying to the Brain Mind Sciences program. After I completed the master's, I moved to Germany for my PhD, and now I do my postdoc in the same institute at the Zero Noise Lab. Yesterday, I was trying to think a little bit about those years in the Brain Mind program, and I was trying to understand what were the things that really helped me later on during my work. And I think I'm actually very thankful about the interdisciplinary part of the program. And there are specific, especially I was trying to remember specific courses that really helped me later on. And here I listed some of them. So for sure, one of the most important contributions, the most important courses for me was the neurophysiology, all of the series on neurophysiology in the first year, the course on cerebral cortex, neuroanatomy, and also the courses on statistics and signal processing. And all of these really are things that I really actively used later on during my PhD and during my postdoc. And also the lab rotations were a great opportunity, which gave me really valuable knowledge. And specifically my lab rotation with Georgia, which really gave me, back then I was working with Panayotis, and he actually really showed me it was the first experiments I saw in non-human primates, electrophysiology, on behavioral training. All of these were very valuable later on for my research. So later on, I moved to, as I said already, for my PhD, and now I will present a small part of what I did in my PhD, which focused on studying the neuronal oscillations of the primate brain using simplified stimuli. Specifically, I studied the neuronal effects of stimulus repetition. And just to give a small introduction to stimulus repetition and why did we study that. So in everyday life and under natural conditions, we typically spend extended periods of time in a given environment surrounded by familiar objects. For example, if you are a monkey, you might look at a given tree several times. or interact with a specific leaf repeatedly within a short periods of time. So how does stimulus repetition affect brain activity? I work with macaque monkeys that are important to advance our knowledge about the human brain. And we also already know that when we present a visual stimulus to a monkey, such as a leaf, spike in activity and oscillatory power increase. And if we average this, the spike in activity over a time window and the frequency activity over the oscillatory power over frequency window, we can actually estimate, we can take a mean value of in response to a single stimulus presentation. So we can have actually a value for the mean spiking and the mean gamma activity. And we can actually then track and see how this activity, this brain activity, changes across stimulus repetition. And as you see here, I think I will activate my pointer. So as you see here, we see that the spike in activity and the gamma show to the different effects. So the spike in activity goes down the more a stimulus is repeated, while gamma band power shows an initial decrease and then increases as a stimulus is repeated. So what do we know already about these effects? We know that from previous studies, we know that immediate repetitions lead to spike decreases and gamma increases. And we know that the effects are stimulus specific and do not transfer to different stimuli. So if we repeat a leaf several times, the neuronal effects will not transfer to a stimulus that's very different from the leaf. And we also know that the effects are present even when we interleave, when we have interleave repetitions of a few stimuli. During my PhD, I focus on these last 2 points, and I try to further investigate these points. And I ask the following 2 questions. So first of all, are there repetition effects when multiple stimuli are repeated interleaved? And do the repetition effects transfer to very similar stimuli? To answer these questions, I performed neural recordings in monkey areas V1 and V2. I performed laminar recordings in both areas, but today I'm going to show you the results from area V1. And the effects were similar across layers, so I'm going to just show you the average response across layers. So we already know, as I said earlier, that repetition effects do not transfer to very different stimuli. But what happens if we present a more similar stimulus? I want to systematically manipulate the similarity between the repeated and the test stimulus. And for that reason, I turned to gratings, which allow precise control over a few stimulus dimensions. And I kept all grading parameters constant, and I only manipulated their orientation. In this way, I could repeat a grading of a given orientation, as you see here, and then use test stimuli of different orientations with varying similarity to the repeated stimulus. So this is now actually schematic representations of how the experiment of one day looked. So in the beginning of the recording session, I had a blog where I presented 18 different gratings, which were presented in a pseudo-random interleaved manner. And this stimuli were actually presented. I presented 10 repetitions per orientation, and I could actually measure the neural responses to each orientation at the beginning of the session. And then one of those orientations was randomly selected, and this orientation was then repeated 90 times in the second block. This is a block that we called conditioning, and at the end, after the conditioning, there was a third block where stimuli of different orientations were presented similarly to the first block. So the first and third block are very similar. So in relation to the conditioning block, the first and the third block can be thought as the pre and the post-conditioning blocks. And this sequence of blocks allowed me to evaluate how conditioning affects the neuronal responses to its orientation. So first, I looked at repetition effects within the first block. And we could confirm that, as people showed before when they presented only very few stimuli, that even when we increase the number of stimuli that we repeat, here we repeated 18 stimuli interleaved, we see that actually spiking activity showed a decrease as a function of the repetition number, while gamma band power increased the stimulus repetition. So we could actually indeed show that we could conclude that there are repetition effects even when multiple stimuli are repeated interleaved. So then we move to the second question, which was about how to do with the stimulus specificity of the effects. And here we compared the estimation of the neuronal activity right before. So actually we estimated the neuronal activity right before and right after the conditioning block. So the conditioning block, as I said earlier, is when we repeat the same stimulus 90 times in a row. So any difference between this and this, the activity at this point and at this point would actually should relate to the conditioning, so to the continuous repetition of one stimulus. We arrange our data as a function of the orientation difference from the conditioned orientation. So an orientation difference close to 0 means high similarity to the condition orientation. And 90 would be the most far away orientation from our conditioned orientation. So as you see here, I plot here the spiking activity for when we say that actually spikes tend to decrease around the condition orientation, which is here at 0. But we show that these effects are not so significant. While if we look at the gamma band activity, we see that for the condition orientation, there is a significant increase. While in the post versus the preconditioning block, which is actually really specific for this condition orientation. So, answering our second question, we say that the repetition effects do not transfer to very similar stimuli in the gamma band activity. So they're actually very specific for this one orientation that was repeated 90 times, at least for the orientation steps that we used in this study, which was 10 degrees. So, of course, if we look maybe for smaller orientation differences, maybe the effect would be then transferred to other stimuli. So we could actually conclude from the study that gamma band plasticity is highly stimulus specific. And we also saw something else that I found very interesting in this study. So we had these beautiful oscillatory peaks in gamma, but we also had some nice peaks in alpha and beta. And we want to see how alpha power in alpha and beta frequencies change within our session. And we actually saw that there is an interesting pattern with alpha and beta, which seems to actually track the predictability of the stimulus. So in the beginning of the session, we say that alpha, beta power, we combine this frequency window is actually pretty low, but the more we present the same stimuli, it slowly increases and then it stabilizes slightly. And then the moment we change our block. Sure. You want to one question to ask?
Yes, then I'd like to clarify a few things actually about the paradigm that you've been using. So first of all, is block 1 same as block 3?
Yes, of course, it's the exact order of stimulus presentation. It's not the same because it's pseudo-random, but yes, they're the same.
Okay, and is this happening during the same day? It's the same day, so it's one session. This is one session,
yes.
Block 1. Okay, and are the stimuli are they static? Are they phase
Yes.
reversing? They're static.
No, they're static. They're static. Yeah.
Okay. Okay, so the recording is done at the same time, and you saw that there was a decrease in spike activity and increase in gamma.
In gamma band power.
Okay. Thanks.
Welcome. Yes, so here with the alpha beta power, we say that actually, so you see here in block 2, which is when we repeat the same stimulus only one orientation, we see that actually in the beginning it's pretty low, but then slowly it actually builds up and it reaches a much higher level than it is in block 1, where we have a little bit less predictability because we have more stimuli that are pseudo-randomly presented, interleaved. And then it's interesting that the moment that we actually change and we present a different orientation, the alpha beta power drops again to this initial level almost, and then it slowly increases again. But again, here that we interleave several orientations, it does not reach the same level as in the conditional block where we have really high predictability. So yeah, so actually, sorry, just to conclude on that, we see here that the alpha beta power in the early visual cortex somehow tracks or is related to the predictability of the stimulus. Unfortunately, our study was not designed to study this effect, the specific effect. But I think something that already some people have shown in the prefrontal cortex with alpha beta power, and I think it's something that it's pretty interesting. So moving forward, I will move now to my current project. And in contrast to what I've shown so far, under natural conditions, we are not passively presented with static stimuli. So for a monkey, this is not a very natural situation. Maybe something like that is actually reflects more the reality of the everyday cognitive processing. So actually, we actively interact and navigate our environment. So my current project aims to bridge the gap between highly controlled paradigms and the complexity of natural behavior. And I'm working as a postdoc, as I said already, at the Xenonoise Lab, which is has two PIs, Marike Solving, who's here with us today, and Martha Havanis. And we study cognitive dynamics in using naturalistic paradigms. And we try to perform the same behavioral paradigms across species. So we use mice, macaques, and humans, which are the three most actually used species in neuroscience. And we try and this allows us, using the same paradigms and the same methods, allows us to actually perform across species comparisons. So as I said already, we want to actually use more naturalistic paradigms, and we want to bring more natural behaviors into the lab. So how can we do that? We actually use virtual reality, and which allows us high experimental control, and while at the same time it allows us to have actually immersive experience. So in our lab, people already have developed a very interesting and actually impressive, to my opinion, setup that allows to use virtual reality for across all these three species. So this is a schematic representation of how the setup looks like. We have a dome. And we use a projector that projects the virtual reality onto a curved mirror, and the curved mirror then projects back to the dome the visual stimulus. So, and we have then the three different species, the mouse, the monkey, and the human, can be sits in within the dome and actually sees the stimuli. So here I have a couple of videos to give you a better understanding of how the setup looks like. On the left, you will see now the mouse. So the mouse is run on a treadmill, and in this way controlling the virtual reality environment, navigates actually the virtual reality. And in this specific task, it has to choose between a target and a distractor. In the middle now you see the monkey performing a similar task. The monkey is actually using a trackball in order to move within the VR. And on the right we have our human who is actually running a similar task, an earlier version of the task. Again, navigating it's in a similar way as the monkey using the trackball. And we have already the previous people already have developed a toolbox that allows to use virtual reality in experiments that allow actually allows to use what we need for experimental use, like event markers, synchronization, and all of this stuff. So if you're ever interested in using something similar, you can have a look on this toolbox and on the paper that's already published. And here you can see the two example stimuli that the subjects can choose. And on the right, you actually see the pathways for the three different species for this task. For the human, the monkey, and the mouse, each trace is actually one trial. And something else that's very important for our lab is that we use rich behavioral tracking. So okay, we actually record the eye movement, so we record the paths in the VR, but we also we take videos of the facial expressions and of hand movements. Here you can see, for example, the monkey using the trackball to navigate in the VR. And we use these rich behavioral-based measurements that we take in order to infer the internal states. If you're interested, you can actually have a look on these publications already from our lab on how we do that. And we ask the question: does brain activity differ across internal states? And how? Here people already have recorded from the monkey areas V1 and V4 using chronic recordings, and also from mice. While the animals are actually performing behavioral tasks in the VR setup. And so we have already intracranial electrophysiology that we can directly compare between the species, but we are actually missing some brain activity from the human subjects. And this is actually now the project that I'm working at the moment. So we're trying to replicate the same setup for human recordings. So I work with several, have a very great team. So we work with people from the Zero Noise Lab, but also with Yorgos Michalareas, who is the MIT professor here in Frankfurt, and Anya Dietrich from the MIT Lab, who really help us to actually replicate now and develop the same exact setup and bring into the MIT to the magnetoencephalography lab. So here on the left, you see how the typical MIT setup looks. Here you see how it is typically, so how the brain recordings can perform when the subject is within the scanner. And here is our dome. So this is a dome. It's a photo before we paint it. So here you see it's a dome, and we have actually cut part on the top of the dome in order to be able to actually bring it close to the MIT helmet. And in this inside part of the dome is where we project our virtual reality. And on the right here, you see how the setup looks when we bring everything into the MIT. We have a support structure, and we have the dome, which is in front of the helmet. And here there are still a few things missing. So we're still developing the setup. The problem with the image is that it's very sensitive to many stuff, and we have to actually find image compatible alternatives for some parts of our equipment. As I said already, we try to have to perform a very rich behavioral recordings, and this is something we are trying to do in the MHA. So we use an image compatible trackball, eye tracking, but also we try now to develop to find a way to actually also perform the video recordings of the facial expressions and of the hand movements, which is something that's typically not done in the MHA. And additionally, something that has not been traditionally done, but that's what we aim to at, is to have a free viewing paradigm. As for our animals, also we use a free viewing tasks. So actually the subject will be allowed to move their eyes around and to navigate in the VR at the same time, which we believe that actually it's important in order to have more naturalistic paradigm. Typically in the MJ especially, it's actually people try to have to not have eye movements because they are very visible in the brain signals. Yeah, so actually when it comes now, just to give you a final overview of what are our plans for the analysis. So we hope that soon we are going to be able to actually really use the setup for the brain recordings, and we are going to record brain activity and MT, and we are going to use the behavior that we are going to track in order to infer internal states, and then group the brain activity into states, so which is going to be like conditions, and then see how the brain activity differs between different internal states. So at the moment we are about to finish building the setup. It has been pretty challenging, but we are very close to finishing the setup actually, and hopefully we would be able to run our first recordings soon. And yes, this is our lab. This is the Zero Noise Lab, and I'm going to finish this presentation with what I would tell myself if I could go back in time before starting a PhD. I don't know, maybe this is interesting for some of the students. So I think if I could go back, I would say that I would really sample as many different methods and research fields as possible during the master's. I think something that I regret that I didn't do during my time in Brain Mind was that I didn't actually. I wish I had done a lab rotation on something more cellular. This was something that I haven't done, and I think it would be interesting. But you have a great opportunity there in this masters to actually really you can try so many different things, and which will help you then really identify the questions and the methods that excite you. And I would really say that before you join any lab for your PhD, really speak with people who have worked there, and start already building your network and collaborate as much as possible. At least to my experience, I think the best way to learn for me has been through collaborations, and choose your collaborators wisely so that you can enjoy your everyday work and you can learn. And I think this is a big part of I don't know, this was a part that I really have enjoyed a lot in science so far, like working with nice people and learning. Yeah, and finally, something that I didn't have is I don't have a plan B, which might be nice and might save you some stress in case that science or research does not work out in the end. Maybe it's nice to have a plan B of what else you would like to do. Yes, but I hope you all enjoy the science and the research, and yeah. I think that's all for me. Thank you for your time and your attention, and I'm open to questions.